Hello everyone, I am Payal Priyadarshini, PG Biotechnology, Kendriya Vidyalaya number 3, Delhi Cant. I welcome you all in the IIT PAL series of learning. So today we will learn about protein synthesis, which is also called as translation. But before moving more detail about the protein synthesis, let us know the answers of this question which I asked in my earlier session. And the first question was, what are introns and exons? So, if we talk about eukaryotic transcription, in case of eukaryotic cell, after transcription of DNA, the RNA transcript is considered as pre-RNA. Pre means precursor RNA. These pre-RNA have exons as well as introns. Introns are considered as non-functional nucleotide sequences, which is also called as intervening sequence. So, introns are present in precursor RNA in eukaryotic cells, which are non-functional in nature and they are not participating in protein synthesis, and these introns are being removed by the process of processing and splicing. And exons are functional nucleotide sequences, which are also called as expressing sequences. So, exons are those part of the RNA which actually participate in protein synthesis, which is also called as expressing sequence. The next question is, what are the function of cap? If you can recall, cap is found only in eukaryotic cell. The messenger RNA of eukaryote have cap at their 5' end, and these caps are synthesized by the unusual binding of guanosine triphosphate at the 5' end of the messenger RNA, and these cap provide the initiation of translation, as there is no any particular sequence in eukaryotic messenger RNA to recognize the start codon. So, these cap helps the ribosome to recognize the initiation site during the protein synthesis, and this cap is present at 5' end of messenger RNA. Cap also increases the stability of messenger RNA. This cap prevent the degradation of messenger RNA by some nuclease enzyme and increase the stability of the messenger RNA. And this cap also influences the removal of introns, as after the formation of pre-messenger RNA in eukaryotes, these introns are removed, and the removal of intron and splicing of exons are influenced by this cap. So, these are the function of cap in eukaryotic cell. The next question is, what do you mean by processing and splicing? If you can recall my last session, processing and splicing is the removal of introns and addition of exons together to form a mature messenger RNA. In case of eukaryotic transcription, after transcription, pre-messenger RNA is being synthesized, which have introns and exons present alternatively. Exons are functional part and introns are non-functional part. These introns are being removed and exons are being added up, and this process is called processing and splicing, which is performed by a small nuclear ribonucleoprotein. The next question which I added over here, and this is an important question, which is the first nucleotide being added during transcription? Here are some options: ribonucleoside triphosphate, ribonucleoside diphosphate, ribonucleoside monophosphate, or any of these. The question means that during transcription which one of these three will be added up first. Either it will be ribonucleoside triphosphate, or it is ribonucleotide triphosphate, or it is ribonucleoside monophosphate. So, answer is ribonucleoside monophosphate, the first precursor, the first nucleotide which will be added up during transcription by RNA polymerase is ribonucleoside monophosphate. It is not diphosphate, neither triphosphate, it is ribonucleoside monophosphate which is added up firstly during the transcription. And the next important question, what are the substrates for transcription? Substrate means the raw material which are being added up during the transcription. It is very much clear that raw materials are nothing, these are the nucleotides, but whether it will be ribonucleoside triphosphate or diphosphate or monophosphate. So, the substrates are ribonucleoside triphosphate. Let us understand this answer. We talk about ribonucleoside triphosphate. Here is the first carbon and nitrogenous bases are being added up at the first number of carbon. It is a ribose sugar, so at the second number of carbon OH is there, at the third number of carbon OH is there, this one is the fourth number of carbon, and here it is the fifth number of carbon, and at the fifth number of carbon there are 3 phosphate. So, this particular nucleotide is called as ribonucleoside triphosphate. You must remember that after the addition of first nucleotide, the next other nucleotides are triphosphate, but during the formation of phosphodiester bond, these 2 phosphate will be removed up to provide the energy for the phosphodiester bond formation, and only ribomonophosphate will remain there in the nucleotide chain of the RNA, but the substrate is always a ribonucleoside triphosphate. So, I hope the answer of this question will be very much clear to you. Now, next move around to know about protein synthesis or translation. Before knowing much about protein synthesis or translation, let us know about what is protein and how does it form. So, protein is synthesized from the RNA, specifically messenger RNA, other 2 RNA, transfer RNA and ribosomal RNA helps indirectly in protein synthesis. So, how this protein is being synthesized, how this information is being flow from DNA to protein, and this flow of information from DNA to protein is called central dogma. So, we have already learned about replication, that is synthesis of DNA from DNA to make 2 daughter copy of DNA from one parental DNA is called replication. Now, the DNA will be transcribed to form RNA and the process is called transcription, and then the messenger RNA specifically will codes for the amino acid to synthesize the protein, and the process is called protein synthesis or translation, and this is how the protein is being synthesized inside the cell. First the DNA will be transcribed in RNA, the process is called transcription, and then the RNA will codes for amino acid to synthesize the protein by the process of protein synthesis or translation, and this is how the protein are being synthesized. Now, what are proteins? So, you must be knowing protein is one of the biomolecule of the cell. Biomolecules are those molecules which are found in living cell and which help in various structural and functional role of the cell. Proteins are organic nitrogenous biomolecule. Organic means they are carbon compound along with the nitrogen. So, all the proteins are organic nitrogenous biomolecule. Proteins are considered as building block of cells. As we know, all the living system, all the living organisms are made up of cells, and these cells are made up of many different types of proteins. And chemically, proteins are polypeptide chain of amino acids. So, if we talk about the chemical nature of protein, the proteins are made up of amino acid. Now, what is this amino acid? Amino acid is also an organic compound with amino, that is NH2 group, and carboxylic, that is COOH group, combines together to form amino acid. And when many amino acid combines together, they will form polypeptide chain, which we can say protein. Now, if we see the structure of amino acid, here is a typical structure of amino acid. You can see in the slide, this is the carboxylic group COOH, this is the amino group NH2, this is the center carbon attached with hydrogen, and this R side chain. This R is variable, and depending on this R, the type of amino acid is considered. So, in most of the amino acid, this amino group and carboxylic group are very much common, only this R group is variable. Now, how do amino acid combines together? As we know, the proteins are made up of amino acid, means when many amino acids combines together, they form the polypeptide chain or protein. But the question is, how the amino acid will combine? Is there any specific role or a specific pathway so that the amino acid can combine? Let us see how the amino acid combines together. So, this is the center carbon, here COOH group is there, hydrogen, R group, R is variable in nature, and here is the amino group. If suppose it is one of the amino acid, say A1, and let us have another amino acid, say it is R dash, and here is the amine group. So, how these two amino acid combines together to form polypeptide chain? So, it is a very simple reaction. The OH of the carboxylic acid of the first amino acid will interact with the hydrogen of amine of the second amino acid. The interaction of these two groups will release the water molecule, and the remaining groups will combine together to form a bond, which bond is called as peptide bond. So, this is the peptide bond which helps in the binding of two different amino acid during protein synthesis. Without the peptide bond, the amino acid cannot join together, cannot bind together, and the protein cannot be synthesized. So, this is how the two different amino acid will combine together with the release of water molecule and the formation of peptide bond. So, you can see over here the OH of acid and the hydrogen of amine of the second amino acid, they will interact together, they will release the water, and the remaining part will combines together with the help of bond which is called as peptide bond, and this peptide bond is covalent in nature. Now, what are the different types of amino acid? Depending on the requirement by the cells, they have been classified into three categories. One is essential amino acid. Essential amino acids are those amino acids which are not synthesized by our cells, and they are being taken up from the food, and they are isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan and valine. Second group is semi-essential amino acid. They are being synthesized by our cells whenever they are being required, and semi-essential amino acid is of two types, arginine and histidine. Third one is non-essential amino acid. Non-essential amino acid means they are not taken up from the food, they are being synthesized by our cells only, and these are alanine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, proline, serine and tyrosine. So, altogether there are 20 standard amino acids which will combine in any numbers to form a polypeptide chain or protein. Now, let us know about the organization of protein structure. Protein structures have different levels. The first one is primary structure. In case of primary structure, the amino acids are combined together to form a linear structure of chain with the help of a bond which is called as peptide bond. This is a peptide bond, and these are different amino acid, say A1, A2, A3, A4, A5 and like that. So, this one is considered as primary structure of protein, which is formed by peptide bond in between the two amino acids. Second level of structure is secondary structure. In case of secondary structure, hydrogen bond is being produced in between the electronegative oxygen and electropositive hydrogen, and this linear structure of polypeptide chain, either they will form a helix or they will form a beta plated sheet. This is called as alpha helix, and the sheet like structure is called beta plated sheet. And this structure is because of the hydrogen bond, as hydrogen bond exists in between electropositive and electronegative atom. The next level of structure is tertiary structure. Tertiary structure means these structure of protein or polypeptide chain will further coiled to form a three-dimensional compact globular structure having alpha helix as well as beta plated sheet with the help of certain more bonds known as ionic bond, hydrophobic bond, van der Waals force, So, these bond is also being added up in the tertiary structure, and the last organization of protein structure is quaternary structure. So, up till tertiary structure there is a single polypeptide chain, but if in this tertiary structure one or more than one polypeptide chain is being added up like this, they will form quaternary protein. So, quaternary structure is the combination of two or more than two polypeptide chain, and to stabilize the protein these bonds are involved over there: ionic bond, hydrophobic bond, van der Waals force, along with hydrogen and peptide bond. So, I hope the level of structure must be clear to you. Moving ahead, let us know how the DNA or messenger RNA codes for protein or amino acid. But the major question is, how many nucleotides are necessary to specify a single amino acid? If we talk about nucleotides, in case of RNA, we have just 4 types of nucleotide. These are ribo, adenosine triphosphate, ribo, uracil triphosphate, ribo, cytosine triphosphate, ribo, guanosine triphosphate. These nucleotides added up during transcription to form the RNA transcript. Now, if we talk about the different types of nucleotide, there are just 4 nucleotides. So, if one nucleotide code for one amino acid, then how many amino acid will be coded by 4 nucleotides? Yes, the answer is very much clear. If one nucleotide code for one amino acid, one nucleotide codes for one amino acid, then 4 nucleotide will code for 4 amino acid. But in a particular protein there are 20 standard amino acid. So, what about rest 16 amino acid? So, this concept was not taken up by the scientist, and then they thought that it might be like 2 nucleotides combines together to code for one amino acid, and accordingly we can have 16 base sequences. If 16 base sequences are there in the pair of 2, they will code for 16 amino acid. If 2 nucleotides combines, here also in the combination of 2 nucleotides only 16 amino acid can be coded up. Again the 4 amino acids are left. So, again this concept was discarded, and then the scientists thought that the combination of 3 nucleotides of RNA combines to code one amino acid, and if so, then there will be 64 base sequences of 3 nucleotides. So, if 64 base sequences are there in the combination of 3, it means the 64 will code for 64 amino acid, but it is not like that. 64 sequences are there in the pair of 3, but they code only for the 20th standard amino acid. So, how it is being done? So, Francis Crick finally concluded that the 3 nucleotides combines to form one codon which will code for one amino acid. So, the codon is a set of bases that encode a single amino acid. Codon is the set of 3 nucleotides, which is also called as triplet codon, and this set of 3 nucleotides which will code for one amino acid is called as codon. Francis Crick and his colleagues confirmed in 1961 that the genetic code indeed is a triplet codon. That means the codon which is going to code for amino acid is a triplet code. That means 3 nucleotide base will combine to code for one amino acid. So, here is the genetic code in the combination of 3 which will code for the different amino acids. So, it is a genetic code which we can also trace out. It is a very easy process how to trace this genetic code. What you have to do is you need to divide the blocks in 4 equal size vertically as well as horizontally. And while writing the sequences in the combination of 3, you need to write U, C, A, G. Here also U, C, A, G, and in this block, in a particular block, you have to write all those 4 sequences U, C, A, G. Here also U, C, A, G, U, C, A, G, U, C, A, G. So, let us start from here U, U, U. So, in this particular block we will be having U, U, U. The next codon will be U, U, C, U, U, C. The next will be U, U, A, U, U, A, and the next will be U, U, G, U, U, G, and like that you can write all the codons in all these blocks. Now, here are some characteristics of genetic code which is an important part. The first is the genetic code consists of a sequences of 4 bases. We have already seen it, there are just only 4 base sequences A, C, G, U. Thymine is not there because messenger RNA is not having thymine. So, genetic code have only 4 base sequences A, G, C and U. The genetic code is a triplet code, that means 3 bases combines together to code for one amino acid. The genetic code is degenerate. Now, what is the meaning of degenerate? Degenerate means one amino acid can be coded by more than one triplet codon, and that is why the 64 codon reduced to 20 standard amino acid. So, degeneracy means one amino acid can be coded by more than one codon except methionine, which have only one codon, that is AUG, which is also considered as a start codon, and tryptophan, which is coded by UGG. So, these two amino acid coded by only one triplet codon, while the rest of the amino acids are coded by more than one triplet codon, and this is what the degeneracy means. The code is comma free. Comma free means it is a continuous sequences of bases which are being coded up. That means, if a messenger RNA is having a particular arrangement of sequences, then during translation or protein synthesis, all the sequences will be read accordingly in the pair of 3 without skipping any base sequence. So, it is read continuously by the ribosome. The next feature of genetic code is it is non-overlapping. Non-overlapping means one codon will code for one amino acid. That means, if AUG is coding for methionine, it cannot code for any another amino acid. Wherever AUG will be there, it will always code for methionine, and this is what the non-overlapping means. The non-overlapping means one triplet codon will code for one amino acid only. Moving to the next features, the reading frame is set by an initiation codon which is usually AUG. So, AUG is considered as start codon which codes for methionine, and the protein synthesis will always be signaled by AUG, which is also called as start codon. The next feature is 3 codons are considered as stop codons, and these are UAG, that is amber, UAA, that is ochre, and UGA, that is opal. So, UAG, UAA and UGA, these 3 codons are considered as stop codon. So, altogether 64 triplet codons are there, 3 are stop codon. So, remaining 61 are considered as sense codon. Sense codon means these 61 codons will code for amino acid, and 3 are stop codon. They are UAG, UGA and UAA. Genetic code is always read in 5 prime to 3 prime. If a messenger RNA is there and it is going to translate it, here it is a 5 end and here is a 3 prime end, and it is always read from 5 prime to 3 prime. So, this will be the direction of translation in case of messenger RNA. The code is universal. Universal means this genetic code is accepted everywhere for all the living system. The important aspect of the genetic code, the occurrence of bubbles in the anticodon. Now, what is the meaning of bubble? Bubble means while pairing of codon with the anticodon of transfer RNA, we will learn about the anticodon in the coming slide. So, anticodon is there in the transfer RNA. When they pair with the codon of the messenger RNA, first two pairing are very much exact, which should be there, but the third pairing may be differ in many cases, because the first two nucleotides give the signal for that particular amino acid. So, third pairing is not very much exactly same as per the order, and this is what we called bubble pairing. You can see over here. So, if suppose it is a messenger RNA and it have CUC as codon, so in transfer RNA in the anticodon loop there is complementary of C is G, complementary of U is A, complementary of C is G. So, GAG anticodon loop will be there in transfer RNA, and accordingly it will code for the leucine. But in bubble pairing, you can see over here in messenger RNA the codon is CUU, and for this CUU anticodon GAG is there. The first pairing is very much correct. The complementary of C is G, the complementary of U is A. Here in the third pairing, the complementary of U should be A, but here it is G. This is not the correct pairing, but because these two pairing can translate, can code for the amino acid, so the third pairing is not the exactly as per the order, and it can code for the leucine, and such type of pairing is called bubble pairing. This is also called as bubble hypothesis. So, I think the characteristics, the features of triplet codon genetic code must be very much clear to you. Moving ahead, the process of translation, how the protein synthesis being started, elongated and then terminated. So, the translation is being completed in different phases. The first one is charging of transfer RNA, second one is initiation of protein synthesis, third one is elongation of protein chain, and fourth one is termination of protein synthesis. The charging of transfer RNA means depending on the anticodon of transfer RNA to the third prime of the transfer RNA, the amino acid will be added up, and the binding of amino acid at the third prime end of transfer RNA is called charging of transfer RNA. After the charging of transfer RNA, the protein synthesis will be initiated with the help of certain initiation factors, and then the protein chain, the polypeptide chain will be elongated with the help of certain elongation factors, and then after the completion of the protein synthesis, the polypeptide chain, the synthesis of the protein will be terminated with the help of certain release factors. So, these are the different steps of protein synthesis, or we can say translation. Now, let us know about the structure of transfer RNA. The transfer RNA is having 75 to 90 nucleotides in their structure. So, transfer RNA is 75 to 90 nucleotide long. They have a clover leaf structure. You have been seeing the structure of leaf of clover. The same structure is there of the transfer RNA. That is why the structure of transfer RNA is also considered as clover leaf model. the transfer RNA have 4 base paired stem separated by 4 different loops. Loop 1, which is also called as T pseudo C loop, loop 2, which is also called as anticodon loop, loop 3, which is also called as variable loop, loop 4, which is also called as dihydrouridine loop, and the stem which is considered as amino acid binding site. In the stem only the amino acid will be binded up. So, you can see the structure over here. This is a typical structure of a transfer RNA which is considered as clover leaf model. This stem is called amino acid acceptor arm. The first loop is dihydrouridine loop, second loop is anticodon loop. This anticodon having the complementary set of nucleotides of the messenger RNA. If suppose a messenger RNA, if it is a messenger RNA and if it have CAC as codon, so in the anticodon the complementary of C is G, complementary of A is U, and complementary of C is again G. So, anticodon loop is the complementary nucleotide sequence in 3 of the messenger RNA codon. The third loop is variable loop. The number of nucleotides can be varied from transfer RNA to transfer RNA. The fourth loop is T pseudo C loop. So, this is a typical clover leaf model of transfer RNA. So, you can see in the slide the structure of transfer RNA. This is the acceptor stem, and at this 3' end the amino acid will be binded up. This one is dihydrouridine loop, this is anticodon loop. These 3 codons are complementary of messenger RNA based sequences. This is a variable loop, the number of nucleotide can be varied from transfer RNA to transfer RNA, and this is T pseudo C loop. So, this is a structure of transfer RNA. Now, let us know the first step of translation, that is protein synthesis, charging of transfer RNA. Before knowing much about the charging of transfer RNA, let me recall the function of all the RNA. As we have learned, there are basically 3 types of RNA: ribosomal RNA, messenger RNA and transfer RNA. Ribosomal RNA helps in the formation of ribosomes, and these ribosomes will help the cell to synthesize the protein. Second one is the messenger RNA. Messenger RNA give the signals for the amino acid during the protein synthesis, and the third one is transfer RNA. Transfer RNA will take up the amino acid as per the signal given by the messenger RNA and bring the amino acid to the ribosome during the protein synthesis. Now, let us move ahead. The charging of transfer RNA. Charging of transfer RNA means addition of amino acid to transfer RNA. Whenever the amino acid is being added up to the transfer RNA, the process is called charging of transfer RNA. The amino acid transfer RNA synthetase enzyme involved in this process. For the charging of transfer RNA, amino acid transfer RNA synthetase enzyme is involved, which helps in the binding of amino acid with the transfer RNA. The process is also called as amino acylation. amino acylation uses energy from ATP hydrolysis. The binding of amino acid with transfer RNA requires energy, and that energy is being taken up by the hydrolysis of ATP, adenosine triphosphate. And the overall reaction is like this: amino acid, transfer RNA and ATP, they all interact with each other in the presence of enzyme, amino acid transfer RNA synthetase enzyme, and they will form amino acid transfer RNA, AMP, because 2-phosphate from the ATP will be removed to get the energy, and that energy will be taken up by the transfer RNA and amino acid to combines together. Now, let us know the steps, how the charging is being done in the cell, how the amino acid is being added up to the transfer RNA. So, the first step is amino acid correspond to anticodon and ATP binds at the specific amino acid transfer RNA synthetase enzyme. You can see in this diagram, this one is an enzyme which help in the charging of transfer RNA, and this enzyme is called amino acid transfer RNA synthetase. This amino acid will combine with amino acid which will correspond to the anticodon of transfer RNA. Here I have taken the anticodon CAA. The codon of CAA will be GUU, and GUU code for valine. So, valine amino acid and ATP, this both will combine with the amino acid transfer RNA synthetase. The next, ATP hydrolyzed to AMP, which joins the amino acid as AMP to form amino acid AMP. In the next step, you can see over here the 2-phosphate of the ATP will be removed, and because of that removal of the phosphate, whatever energy is being liberated, that energy is being utilized in the binding of amino acid with the AMP, adenosine monophosphate, and the binding of these two in the amino acid transfer RNA synthetase enzyme gives amino acid AMP enzyme. The next step is transfer RNA binds to enzyme. You can see over here, this is a transfer RNA without any amino acid, which is called as uncharged transfer RNA. Now, this uncharged transfer RNA will bind with the amino acid AMP enzyme. After this, the next step, transfer of amino acid to transfer RNA. Now, this amino acid will combine with the 3' end of the uncharged transfer RNA, and while the formation of bond between amino acid and uncharged transfer RNA, this AMP will be removed up, and these amino acid which are being combined added up at the 3' end of the transfer RNA is also released as amino acid transfer RNA or charged transfer RNA. So, this is how uncharged transfer RNA is being converted into charged transfer RNA with the help of enzyme amino acid transfer RNA synthetase. Let us recall all the concept. Amino acid transfer RNA synthetase will combine with amino acid and ATP molecule. After the combination of ATP and amino acid, the 2-phosphate will be removed with the release of energy, and that energy will help the binding of valine and AMP. After this, the uncharged transfer RNA will be binded with the enzyme, and after the binding of uncharged transfer RNA, this AMP will be removed, the amino acid will be added up at the 3' end of the uncharged transfer RNA, and after the binding of amino acid with the transfer RNA, the transfer RNA become charged and the charged transfer RNA will be released. So, this is how the charged transfer RNA is being produced by the amino acid transfer RNA synthetase enzyme. Now, the next step of protein synthesis is initiation of protein, means how the synthesis of protein is being initiated by the cells. So, initiation of protein synthesis requires ribosomes, a specific initiator charged transfer RNA, messenger RNA, GTP, that is guanosine triphosphate as the energy source, and initiation factors. So, these are the primary requirements for the initiation of protein synthesis: ribosomes, a specific initiator charge transfer RNA, messenger RNA, GTP molecule, and initiation factors. Let us know about this. If we talk about ribosome, in case of bacteria or prokaryotes, the ribosomes have two subunits, larger subunits and smaller subunits. Larger subunit is 50S, S stands for sedimentation coefficient or Sedberg unit, and a small subunit is of 30S. When smaller subunit and larger subunits combines together, they will form 70S ribosome. These subunits contains 23S, 5S and 16S ribosomal RNA in them, and these ribosomes helps in reading the codon of messenger RNA during protein synthesis. These ribosomes will read the codon of messenger RNA, will give the signal for the amino acid, and the protein will be synthesized. And if we talk about eukaryotic ribosomes, as that of the prokaryotic ribosome, the eukaryotic ribosome also have two subunits, larger subunits with 16S and smaller subunits with 15S, and the combination of these two will give the 80S ribosome. These ribosomes contain 18S, 5.8S, 28S and 5S ribosomal RNA, and these ribosomes will read the codon of messenger RNA during the protein synthesis. So, you can see the structure of prokaryotic and eukaryotic ribosomes. Ribosome is a very important part of protein synthesis, which reads the codon of messenger RNA, give the signal to the transfer RNA, and the transfer RNA, depending on its anticodon, will bring that particular amino acid to the ribosome to synthesize the protein. So, the next steps of protein synthesis we will learn in our next session. Before completing this session, let us think over the question which I will give. The answer of this question will be given in my next session, and these questions are: what do you mean by protein synthesis? Second question is: which enzyme helps in charging of transfer RNA? Third question is: which codon is called initiation codon? If you can recall all those concepts which we learned today, you will be able to answer this question. Next question is: what do you mean by stop codon? what are the stop codon and what is the function of a stop codon in protein synthesis. And the last question, what are the role of ribosomes during protein synthesis? What are the different function is being performed by the ribosome during protein synthesis? With the answer of this question, we will meet in my next session with a continuation of protein synthesis. Thank you very much.